Thanks a lot for the invitation. Yeah, I will try to talk how one can use DNA as a construction material at nanoscale. So yeah, this is not a typical audience I talk to, so I really hope that you will find something interesting and maybe even useful from this talk. Yeah, we humans are very well equipped for building and shaping materials at the millimeter and centimeter scale. We can simply use our hands, right? So if we go smaller to millimeter scales, things become a bit more complicated, but yeah, we can use tools like tweezers to manipulate small objects. So if you go even to a smaller scale like micrometers and nanometers, and you want to build something from molecules, proteins, metal nanoparticles, quantum dots, and so on, it becomes really, really challenging. And the reason for this is what people sometimes refer to as a tiny hands problem. Simply, we don't have such a small hands to arrange things in the order we would like them to be. And one possible solution for this problem is self-assembly. And for us, self-assembly is usually defined as a spontaneous organization of components into desired structures through specific local interactions among components themselves. So that's a bit of a technical definition, but it's important, and it should be clear why it's important later. Yeah, so basically you can imagine that you just put your components together, give them some time, and they will come and form a structure which you wish them to form. Self-assembly is not very visible in our everyday life, right? Usually it takes some work to assemble things together. So yeah, I have a lot of these Lego pieces at home, and they never self-assemble them into the structures they have been previously. Well, but if you look at nature, nature uses self-assembly all the time, and actually across all scales. So we are essentially objects built by self-assembly process. So cells, which are in millimeter and micrometer scales, are built by self-assembly. And if you look even in the smallest scale, like nanometer scales, life is essentially enabled by self-assembled molecular devices, which are capable of performing rather complicated tasks. Just a few examples. So your genetic code is read out by a protein called RNA polymerase, and what it does, it moves along with your DNA, it reads the genetic code and translates it into another molecule called RNA. Photosynthesis, which is again essentially one of the processes which enables life on Earth, which converts the solar energy into a chemical energy. And it's also enabled by one of these proteins which can capture the light and then translate it into the chemical fuel. And that's another example. So all of us are actually driven by these small guys called Atp Synthetases. So what they do, they create a chemical fuel which is then used by cells in all other chemical processes. So let me show you some more visual examples. Let me take this out. I'll have to find a way to start the video. So that's a illustration of ATP synthetase. What it does, it's yeah, as I was mentioning, majority of biological processes inside the cells are powered by this molecule called ATP. So what happens that there is this chain of phosphates. There are 3 phosphates, and if you break one, an energy is released, and then this energy can be used for some other purposes. As you can see, when the energy is released, a lot of material is there is a lot of leftover, and it would be stupid to waste it. So what this ATP synthetase does, it converts it back to the chemical fuel. And that's how it looks. It's a kind of a rotary-like motor which uses the gradient of hydrogen ions to power it. And when it rotates, it's resynthesize this used fuel back to the useful fuel. And yeah, as I was mentioning, all of us are essentially powered by this. And during the day, we recycle about half of our weight of ATP. So yeah, you can also say that we are ATP recycling machines. One of the guys which is powered by this ATP is a motor protein called kinesin. And what it does inside your body, it essentially moves things around. And the process is really this mechanical. It has two legs, it has two arms, it can grab its cargo, and then there are tracks inside your cells along which it can move and then deliver the cargo from the place where some materials are synthesized to the place where these materials are later used. Oh, sorry, now I jumped too much. So yeah, I mean, when we look at this kind of objects, we start to think, can we build something similar, right? And just from kind of building point of view, there are several prerequisites which you might consider as the path towards functionality. So one of them seems that in order to build a functional nanoscaled machinery, one thing you need is to control the structure, because the structure of this object is very well defined. And in biology, there is a kind of fundamental underlying rule that the structure very often defines function. Right? So, and another thing you notice that a lot of functionality of these things comes from some kind of structural reconfiguration. So they move, and this process of movement is essential to what they do. Right? So if you want to mimic something like this, we have to find a way how do we build defined structures and how we make the structures change their configuration in a controlled way. Right? And it turns out that DNA is actually very good material for building things, and also building things which can change their spatial configuration in controllable way. Okay, let's get a bit more technical. What is DNA? DNA, single-stranded DNA is essentially a polymer. So it's just a long molecule of repetitive similar units. So it has a backbone here to which 4 different types of so-called bases are attached. Now you don't need to remember the precise chemical structure of DNA for the rest of the talk. But what is useful to remember that these bases inside DNA can bind to each other in a very specific way. So so-called G base binds to the C base and A base binds to the T base. So if you have 2 DNA strands on which for every A base there is a corresponding T and for every C there is a corresponding G, well, they can bind to each other and then they form this classical right-handed double helical DNA structure. Right. So DNA has a number of very useful material properties. So first of all, you can synthesize DNA of desired length and desired sequence. This is something which is not trivial with other polymers. Right. You can cut DNA at desired places and you can join it together just using biotechnology toolbox. Right. You can introduce all kinds of chemical modifications inside DNA. And also double-stranded DNA, it's a relatively rigid on a nanoscale object. But if you take a single-stranded DNA, it's very flexible. So it gives you some kind of space how to play with the material properties of it. Yeah, just for reference, a diameter of a double-stranded DNA is around 2 nanometers. So it's a truly nanoscale object. So yeah, well, usually when people think about DNA, they think about double helix, but this kind of base pairing rules inside DNA, it gives you possibility to program not only how 2 DNA strands bind to each other, but how 3, 4, and more DNA strands interact with each other. So, and yeah, and that's why I kind of brought this big text for the definition of a self-assembly, because the specific and local interactions are very important aspects. And with the DNA, you can control and program those properties relatively easy. So the field which uses DNA as a construction material is called DNA nanotechnology. It has been going through a very rapid expansion in the last few decades. It's a somewhat updated slide ending at, well, 2012, but the growth continues. So yeah, we learned how to build rather complex objects using DNA, and I will give a few examples later. But what I want to do now is actually to go here and to tell the origin story of the field, because it's quite interesting and somehow inspirational. So it all started with this guy. Now it's a long slide. Let me see. Yeah, it's a long slide. Anyway, it started with a guy called Nedrian Zeman. Nedrian Zeman originally was a crystallographer. And if you are a crystallographer, what you do, you grow crystals, and from the crystals, you want to determine the structure of a certain molecules. His problem was that his crystals didn't want to grow. And then if you don't have a crystals, you are not much of a crystallographer because you don't have your objects to study. So how he tells it himself, that one evening he was rather desperate and went to a campus bar to have a beer. And then this picture from Escher called Depths came to his mind. And yeah, I don't know what majority of people see in it, probably just a bunch of flying fish. But he saw here a crystal. So he saw a crystal of biomaterial. And then he thought, okay, maybe instead of growing directly crystals of my molecules of interest, I can build some kind of scaffold or a frame to which I can then attach my molecules of interest and then yeah, do my crystallography magic on them, right? So yeah, this is also, if you look at this depth, basically requirement is that if you have an object with a certain polarity and valency, meaning that, okay, there is a fish which has a head and a fin, and fish binds to each other, fin goes to the head, tail goes to the head, and then left fin to the right fin, and so on and so forth. So yeah, he essentially wanted to build an artificial crystal on molecular scale. He wrote a theoretical paper in '82 about the underlying principle, how this should be made. That's image taken from his original paper. It's a bit crappy because it's quite old, but that's a bit better illustration of what his goal was. Now, if you want to build a lattice, what you notice that the lattice, basically, whatever lattice you want to build, it has junctions, right? And luckily at that time, a junction structure made out of DNA was already known. So it was known that DNA can form not only just a double helix, which is just a straight rod-like object, it can also form a four-arm junction called a holiday junction. It's a naturally occurring structure. So they were able to fabricate artificial holiday junction out of DNA pretty fast after the original idea. It took them just 1 year. They published their Nature paper, so they were quite happy. And then, well, it's relatively straightforward, so you should take your junction and then try to assemble them in a lattice, right? So to do this, you just expand a short regions here with a single-stranded DNA, and then you can program how different ends bind to each other. So it seems relatively easy, but actually it never worked in practice. So what they managed to do, they built something like that. It's a discrete object, because building a discrete object is much more straightforward and easier than building a large lattice, either in 2 dimensions or in 3 dimensions. Right? So actually they succeeded in a while to build a two-dimensional lattice out of DNA. So that's an AFM image of a small pieces of a DNA, but you can see a rather regular arrangement of features. And the important thing here is the scale. So the separation between these two features is in the order of 10 of nanometers. And at that time, it was impossible to structure materials, any materials, in a periodic fashion with this kind of spatial precision. But again, it took them around 17 years from the original idea just to go to the two-dimensional lattices. And at the end, they actually managed to build a three-dimensional crystals out of DNA. So it's just DNA. There are no other molecules attached to it, but those are already rather large objects for us. So you can almost see them with your eye, but this is an optical microscope image. So it's a crystal made out of DNA. And it took them around 30 years to go from the original idea to the realizing it experimentally. So yeah, why I'm telling this kind of origin story is just to remind us that it takes time to go from an idea to building something in practice, especially if you try to build something which has never been built before. It's always good to look for the inspirations outside your immediate field of expertise. So I don't know if Ned might not be familiar with the Escher drawings. I might not be doing what I'm doing now. And yeah, it's always good idea to go for a beer now and then because something interesting might come to your head. Well, meanwhile, there was another person who made critical contribution to the field called Paul Rosamund. He was in Caltech. So he invented an approach that allows to build DNA structure in a relatively simple way with a high yield, and the whole process is much more robust than before. So instead of building objects through the combination of small DNA pieces, what he proposed is that let's take a long single-stranded DNA. So it's usually we call it scaffold. It comes from a virus. It's a genome of a virus. It's not a human virus. It's a virus that infects bacteria, so it's safe to work with. And then he designed a bunch of short DNA strands, and they are designed in such a way that they can bind this long DNA piece in two distant places and then bring it together. And when they do it, they kind of staple it at the defined positions and then force the whole long guy to fold into the structure which you designed it in advance. Right, so this is a technique called DNA origami typically. Yeah, and it combines this long strand and around 200 short synthetic DNA strands together. So that's how kind of the process works. You take your DNA strands, you mix them together, you add some salts, you heat your solution up, and as you cool them down, these DNA strands will come together, bind into the places they should bind, and then form the structure which you wish them to form. Good. So Paul already showed in his initial work multiple type of structures which can be made, like rectangle, star, smiley face, a bit more complex rectangles and so triangles and so on. A typical scale here is in the order of 100 nanometers. So this is a truly nanoscale thing. So it took people some time to realize how this can be translated into the 3D, and in 2009 a group in Harvard demonstrated that you can use the same approach to build a 3-dimensional objects completely out of DNA. So these are electron images of the experimentally realized objects. One of the guys which was behind this 3-dimensional work, his name is Tim Liddell. He's a good friend of mine. So he has some interest in an architecture. So he thought that, okay, can we build now a structure, kind of tensegrity-like structures? And tensegrity structures, I probably don't have to tell what those are, but just in case. So you have a combination of objects which are beams under compressions and cables under expansion. So so here just 3 wooden beams and some ropes, and the structure is quite stable. It's held together by this combination of mechanical stress and expansion. So he wanted to realize something like this completely out of DNA just for fun, but not only for fun, because tensegrity gives you kind of a possibility to fill the volume by using very little material, right? So he kind of mimicked the structure just using 3 bundles of double-stranded DNA, and then these ropes here are also made out of DNA, but instead of double-stranded, it's now a single-stranded DNA. So single-stranded DNA, it's a flexible kind of polymer, really a rope-like, so you can stretch it relatively easy. Yeah, that's his experimental image. It's not very visible, but with some imagination, you can see here these 3 bundles together. Yeah. So Tim also comes from Munich, and Munich has this great example of a structure built using similar approaches. So it's an old Munich Olympic Stadium. There are these beams standing here and then cables which are holding a beautiful roof over the stadium. Yeah, so now these days we can actually build very, very complex object out of a DNA. So this is just illustrating images, but we can build a long rods out of them just by binding multiple origami structures together. We can build banded structures like this. We can build 3-dimensional, very complex objects like this ball. And this is an interesting example because it's not possible to build something like this at nanoscale using any other fabrication approach. Right, so you cannot use lithography, you cannot use chemical synthesis. Basically, DNA origami is the only technique which allows you to realize structures of such complexity. Yeah, just to show you some real examples. So this is from the group of Henrik Dietz, again in Munich. So recently, at the end of last year, they published a collection of multiple DNA origami objects and did very careful structural analysis of them. So those are real reconstruction of microscopy data. That's really how the structures look like, right? So there are some barrels which are empty inside and all kind of blocks. These structures they call the, this they call the hinge. And so on and so forth. And again, typical dimension of these guys is several tens of nanometers. Good. So let me tell you how our manufacturing process works. So we start with a computer-aided design process. So we first think about what kind of structure we want to build. And let's say if I want something like a cross, there is a computer program which allows me to design it. What the output of computer program is basically a DNA sequences which I need to build this object. So I take an Excel file which has around 200 DNA sequences in it. I send it to a company, and then company sends me back my DNA strands in this kind of plates. I mix them together with this long scaffold strand which comes from a virus. I put them into a machine which heats them up and cools them down, and then I go to a microscope to check if the result of my manufacturing is actually what I expected it to be. So that's how the design looks on my computer screen. So this is essentially our blueprint of the structure. So if you zoom it in, you see how all the DNA strands, where do they go, and what is the sequence of these DNA pieces. Yeah, that's what I get from the computer program. It's basically the real sequence of each DNA strand. When I send it to company sends me back this piece of plastic. So it's a 96, so-called 96 well plate. In each of these positions, there is a small drop of water with the DNA material inside. So I mix it together. I put it inside this kind of very expensive cooking machine, which heat it up, cools it down. And then, yeah, I go to this kind of microscope. It's an electron microscope, which allows me to see that what I get is actually what I designed to get out. Okay. So there is another very important aspect of this DNA origami object, which comes from the fact that each of the short DNA pieces inside the structure, it's unique in sequence and position, right? So then I know which DNA strand will be on the left eyebrow of my smiley face and which one gonna be on the right one, which gonna be on the lip, and so on. So then I can modify DNA strands in the specific positions so they can act as a binding sites for attaching something else. So for example, it's possible to attach proteins, metal nanoparticles, quantum dots, and so on and so forth. So this is an object of 24 proteins, and they are arranged in this structure. The distance between proteins is around 6 nanometers. So again, there is no other technique which allows me to arrange nanoscale materials with such precision. So essentially, DNA origami allows you to assemble what you want, where you want, and when you want. Right? That's putting it in a very simplistic terms, maybe in a more academic ones. So it's a technique which allows you to build complex structures. Hopefully, the structures will have some novel properties and functionality, and it's also possible to implement stimuli response into them. Now, to get a novel property from the structure, there should be some kind of interactions from your individual components, right? Otherwise, it doesn't matter if they are together in a complex pattern, if they don't interact with each other or with environment in some kind of structure-specific manner, right? So there should be some emerging property coming from a structure. Otherwise, there is no point building them. They look nice, but they don't do anything new, right? So an example of the objects which interact with each other quite well is metal nanoparticles. So metal nanoparticles, if you have two metal nanoparticles far apart or close together, if you have them close together because of interactions, their optical responses might be very, very different than when they are far apart, right? So, and actually people are now very skilled in building all kind of metal nanoparticles, variously shaped, rather complex ones. So this is just a frame, empty inside. But for a long time, there was a problem. How do we arrange this nanoparticle into a specific structures, and hopefully, how do we get new properties coming from this precise arrangement? And this is actually one of the reasons why I'm in Aalto doing what I do, because around 10 years ago, we decided that, okay, let's build a structure using the DNA origami approach. And the structure should have a certain prerequisites. So it should show a novel property which comes from interaction of precisely arranged metal nanoparticles, right? And also, to make things a bit more complicated, let's build a structure which is truly three-dimensional. So we built this kind of helices of metal nanoparticles. There are 9 particles arranged either in a right-handed or a left-handed helix. So these structures are so-called chiral, right? So they're either right-handed helix or a left-handed helix. A word chiral, well, classical example of a chiral object is our hand, and actually the word chiral comes from the Greek hair, which is stands for a hand. And the chiral objects, usually they have a quite interesting optical properties, so they can interact with a light, left or right circularly polarized light in a different manner. So you don't need to kind of remember too much about it. It's just that if you have a chiral object which is right-handed, it's going to have different optical properties than if you have a chiral object which is left-handed. So yeah, we build this kind of helices. They assemble with a pretty good yields. And then we characterize the optical responses, and basically we got what we expected. So they showed a specific optical response from our experiment, and there was a very, very good correspondence to a theory. So this match between theory and experiment was actually a very important aspect, because now what you could do, you could design your nanostructures and predict their properties just using calculational approaches. And then when you do experiments, you basically get what you expect to get. So now it's instead of maybe building every time a different structure, you can predict and design the properties using calculational tools. Good. So yeah, it was a nice work demonstrating, let's say, the link between structure and a property. So if you build a left or right-handed helix out of nanoparticle, you get a specific optical response. And back then, this kind of optical responses were still somewhat exotic ones. Right. So, but obviously the structure is static. So after you build it, you are fixed essentially with its responses and with its properties. So we spent a lot of time trying to figure out how you can also implement stimuli response into it, right? So how you can build a structure which can switch, and by switching its configuration, it also has a different property. So, and one way to do it is to take a DNA origami which is flexible. So now it's just two bundles of DNA origami joined with a flexible link so they can rotate. We still add metal nanoparticles, but this time it's not a spherical particles, it's a rod-like particles. It's still a chiral object. So this is a right-handed arrangement of the metal particles. And then to make them stimuli responsive, we implement a small molecular locks inside them. So these locks can react to a stimuli, for example, a presence of a specific molecule or a light. And when it reacts, it gets open, and then you can sometimes close it back again. Right. So again, that's how the structures look under the microscope, and that's how they look after you assemble the gold nanorods on top of them. So again, note the scale. So these rods are around 60 nanometers wide and 10 nanometers in diameter. And yeah, basically, for example, you can make the small molecular locks light responsive. So if you shine light which is in UV region, it gets open. And if you shine a light in a visible region, it gets back closed. And you can do this kind of switching back and forth multiple times. Right. But this is an example of very kind of straightforward thinking. How do I implement stimuli response? So I want stimuli response. What I do, I take a stimuli responsive element and plug it inside my structure. There is a bit of a different way how you can do it. So instead of plugging a stimuli responsive element inside your structure, what you can do, you can put your structure inside a stimuli responsive environment. So now your structure is not responsive to the stimuli itself, but it is in the environment which is responsive to the stimuli. And then the structure is responsive to the change of the environment. So we were able to build this kind of origami constructs which change when you shine a light upon them, but the objects themselves are not responsive to light at all. There are no light responsive modifications, and the response is incorporated through environment. But otherwise they do basically the same thing. They switch, and you can switch them back and forth multiple times. Good. I think I'm kind of starting to run late, but let me then talk a bit about functionality. So that's the last thing in this kind of logical chain of properties. And by functionality, I mean quality of being well suited to a specific purpose. So it's going to get a bit technical, but I will show a few examples how they can be used, this kind of structures for nanophotonics, drug delivery, and biosensing, very, very briefly. So they are potentially very useful for nanophotonic purposes, and it comes from the fact that if you have a kind of arrangement of metal particles close together, if you put a molecule inside the gap between these particles, you can make your molecule very, very, very bright, right? So that's how it actually looks in the microscope image. There are two big particles, DNA origami template between them. So, and you can make this molecule so bright that you can actually see a single molecule using your mobile phone. So you don't need expensive optical setups or anything like this. So basically, each of us is now carrying a good enough detector to be able to see a single molecule. And of course, a single molecule is its ultimate limit. You cannot see better than this, right? And this is just a commercial smartphone. It was published 2 weeks ago. It's a Huawei P20. It's not even the most advanced ones. So yeah, but if you build this kind of construct, you can get a very, very sensitive optical device out of it. You can also build the structures which are potentially useful for a drug delivery, and this is kind of one of my favorite examples in this whole business. So it's a barrel-like DNA origami construct, which is loaded inside with an antibodies. And then it's also locked together with the molecular locks, which can react and open only when they find a cells of a specific type. So let's say if you want to find a cells which are harmful, like cancer cells, when you add to your body or to your experimental conditions this kind of closed origami-based barrels, they will float around. When they find the cancer cells, they will understand, well, not understand, but they will react to the presence of the cancer cell. This barrel will get open and it will expose the cancer cells to the antibodies or a drug which is loaded inside. And when it exposes the cells to these antibodies, it affects what the cell can do later. For example, it can kind of tell the cell, okay, now you should die, right? So it implements multiple functionality. So it implements recognition. So it gets to do its trick only if a specific cells are present, and then it can also affect the cell behavior. And ourselves, we have been working a lot with the biosensing applications. So again, if you can implement an element in your structure which are somehow responsive to the presence of a specific molecular targets, you can switch your structure between several configurations. Each configuration has very different optical response. So you get kind of concentration dependent optical readout, which tells you how many molecules of interest might be present in your solutions. Right. So yeah, where the field is now, it's still at the very, very early stage. So there are multiple demonstrations of kind of promising proof of concept studies, but there are still not so-called killer applications, right? This technology is not yet used in any. Oh, well, there are some commercial products, but they are very, very niche-like, right? So despite, there is a lot of potential, but we are still figuring out what this kind of approach for building nanostructures is good for. Interestingly, this initial idea of building, let's say, a scaffold for arranging other biomolecules, it's never worked out. And it's actually became kind of irrelevant. So there are other techniques now besides X-ray spectroscopy, which allows you to do your structural studies. So even though the initial idea is never worked out, it gave it kind of allowed many other research directions to emerge out of it. And of course, if you look and compare the DNA origami approach to the natural functional nanostructures, so we are approaching maybe structural complexity, which is similar to the natural objects. But in terms of functionality, we are very, very, very far. So, and it, well, kind of not to get completely desperate and frustrated, it's important to remember that nature had billions of years to optimize its design principle and figure out how to implement this kind of functionality. Right. So, yeah. Just a final note. So it's not actually clear if kind of the DNA gonna be the material that we use for building nanostructures in the future, right? So it might not be the best for this. And it's really kind of a unnatural way to do it out of DNA. So nature doesn't use DNA to build its functional machinery. It uses proteins, right? But we don't understand proteins well enough to build artificial structures with a desired functionality. But we do understand DNA well enough to build a complex structure. So at the moment, it's not the best, but it's the best understood material so far. Good. So that's all I wanted to tell, and thank you very much for your attention.